Good afternoon. We're going to go ahead and get started with the dbGaP submission webinar. If there are questions that we don't get to, we'll put them on the FTP site after the webinar. They should be in the same FTP directory as the slides. Slides that are produced by NCBI are open to use by anybody, and so please feel free to use them in educational pursuits. Now I'm going to turn the presentation over to Mike Fiolo. Mike is the head of the dbGaP group at NCBI, and he's going to tell you about submission to dbGaP. This webinar is meant primarily for folks that work on the NIH campus or for the intramural research program, but about 95% of the webinar will be applicable to everyone interested in submitting to dbGaP. Here's Mike.
Thank you, everybody, for coming today. First of all, I want to talk about what dbGaP is and what it isn't, how to get your study registered, and how to format the data for submission, and then finally how to upload the data. What's not covered today is how to submit SRA data to dbGaP controlled access. That'll be in upcoming webinars. Just to orientate everybody, the dbGaP is part of the National Center of Biotechnology Information. I think most of you know NCBI from PubMed and PubMed Central. You may be familiar with our Genome browsers. NCBI actually covers from genome all the way to medicine. PubMed certainly does that. When I came to NCBI back in 2000, these were the primary databases in the NCBI, and I think most of you know dbSNP is a variation database, EntreGene, RefSeq is a curated sequence database, GenBank is the original kind of molecular database at NCBI, contains many sequences from mostly Sanger-based sequencing. GEO is a database for primarily array data from expression array data. And then OMIN is the online Mendelian inheritance in man. So it's an online version of the Johns Hopkins curated resource. So shortly after I arrived, I worked on dbMHC and dbSNP originally. The trace archive was coming online. This was to take actual data from mostly Sanger sequencing back then. And then in about 2006, we started building what's called dbGaP. And this was to house the newly set of data for the GWAS studies that were coming in right after HapMap. And then we built a pGene which shows association results for those data. Shortly thereafter, several other databases were developed here at NCBI. The SRA, which is Sequence Read Archive, to take next-gen sequencing information and compress it and store and archive it. dbVar is a database of structural variation, much like dbSNP. ClinVar, which is similar to OMIM, but not so much curated, but it's not textual based like OMIM is, but it has more structured elements to it. And then MedGen and GTR, the genetic testing registry. This slide is mostly just to orientate you kind of on NCBI. To get into the meat of the talk, the purpose of dbGaP primarily is a permanent archive. It provides public accessions that can be referenced in future manuscripts referencing the data. We have several data objects that are versioned so that they can be updated, data can be redacted or appended to it as new data becomes available. There's two modes of access for dbGaP. One is a public display of summary level data and study metadata, and the other is a controlled access for individual level data. There's several different types of broad types of data in dbGaP. One is phenotypic data, and this is somewhat unique to dbGaP, especially here at NCBI, where we take patient-level demographic, clinical, biomarker exposure variables, but we can also take images of people as part of the phenotype broad data type. And then we have study documents, which are primarily questionnaires or data collection instruments or protocols on how data was collected. The third class is a class of data broadly referred to as molecular data, but this originally was, for the GWAS studies, array-based SNP genotypes. And then more recently, in the last few years, we started to see a lot more other types of molecular data, such as imputed genotypes, sequence-derived genotypes in the form of VCF files or MAF files, which are SNVs, tumor normal comparisons. And then there's increasingly we're seeing next-gen sequencing submitted to dbGaP studies along with the phenotype and study documents. And this is all kind of housed in the SRA, the Sequence Read Archive here at NCBI, but it's the protected mode of the Sequence Read Archive, so it's behind all the firewalls that dbGaP has also. So they broker the Sequence Read Archive submission, and that's kind of why there'll be another webinar talking about SRA submission in the future. That archive actually can take DNA and RNA sequencing, methylation, next-gen expression type assays, and then dbGaP also takes association analysis results, which are the p-values from the analysis of phenotype to genotype. The dbGaP is actually governed by the GDS policy, which is a genomic data sharing policy. And this is a URL to their website. And I thought I would just show that and actually bring it up live to point out a few aspects of the GDS website. One is you can go and read about the policy if you haven't already. And you can see the oversight committees. But I think a lot of you would probably be most interested in the researchers section, and I advise you to read this if you need to or want to submit to dbGaP. And I'll just point out when you click on researchers, another tab comes up called institutional certifications. Now these are the institutional certifications that will be signed by the submitters that agree that it's appropriate for the data to be archived and distributed through dbGaP. So I advise you to look at that also. So I'll just close that down now and go back to the slides. Getting into how data comes into dbGaP in the broad sense. This is a typical example. Often we have the phenotypic and study metadata information come in from a cohort or principal investigator that has access to this samples and actually the people behind the samples and their information. And then often we see that studies have a sequencing center, and this might be the Broad, Baylor, New York Genome Center, others that are submitting data on behalf of the PI and will be housed in what we call a study in dbGaP. I'd like to point to the dbGaP homepage and just show that if you're new and to the submitting to dbGaP, that the first place to start is how to submit. And if you click on that link, you'll actually go to a flow chart, which is kind of a 1,000-foot view of the process. And I'll just point out this flow chart is set up in kind of 9 sections. There's 3 on the vertical axis of these are different roles. So the principal investigator, which I'll call PI from now on, and then the Genomic Program Administrator, which is the IC, so the institute or center's liaison to dbGaP and manages and the administrator of registering studies in dbGaP and certifying the submission is correct and really administers the whole part of the submission registration process. As you'll see, the dbGaP is really not involved in this part of the process. On the horizontal axis, we see study registration, data submission, and study release. The process starts when a principal investigator wants to or needs to submit data to dbGaP. The first question is the study NIH funded? And if yes, then you need to contact your NIH program officer or Genomics Program Administrator if you're intramural, which I think a lot of you are. If the study data was not produced by NIH funds, then you want to go to this link here and follow NIH guidance. But this process really is a way for a GPA at an IC to sponsor the non-NIH funded studies and connect these PIs with the GPAs when appropriate. And it's up to the ICs to decide whether that is or not. The Genomics Program Administrator, GPA, starts the registration in the system. It's a system that it's an online system where they can enter study details. And this process has kind of two modes. One where the GPA enters everything and completes the registration without involving the PI. But the PI does need to collaborate and provide the information that needs to go into the registration. Or the GPA can actually invite the PI to upload to the system. And it's an online system. It's not, it's fairly intuitive. Although that's up to debate, I guess. But so you may or may not actually work with the registration system. So I'm not going to really show that part of the process. But going through the process, at some point the registration is completed, and this involves having the institutional certification and the consent categories for the patients set up. Once the registration completes, there's an invitation that goes out to the PI, and they are invited to upload to our submission portal, and I'll show more of that later. And then the PI would format their data into concordance with our submission guide, and then submit those data. Now, as I showed on that other slide, the phenotype data might come in from one group, and the molecular data comes in from another group. At this point, the data submission has happened, and dbGaP begins kind of their process is to just start processing the submitted data. We assign sessions, and we create a preview site. We do some data QC checks on the data, and then once that's done, we provide the preview site for the PI to look at what would be the public data, summary level data, for their approval to make sure that the study looks appropriate on the public release. And then if all goes well and the QC passes, although there could be iterations here or here, we would release the study. So that's kind of the broad overview of the process. Now I'll get into, I did want to point out here, if you click on this link here, you get a list of the Genomics Program Administrators. This list is longer than what I'm showing here on this slide, but your IC or the IC that's funding you should be shown on here. So now I want to talk more about what's involved in getting the data ready to be submitted in this process here. And so the first thing I'd like to say is that in dbGaP, we have set up our submission formats very simply as rectangular tables. And we have some core tables that are required, and we do this in a modular fashion so that one can be updated without having to touch the other ones. But just to give you an overview of kind of what we're expecting is one of the core files is a listing of all the subjects and their consent categories. Now, a consent category is defined by the data use limitations that were originally agreed upon by the study participants when they sign their informed consent. This acts as a master list of study participants, and we have a way that you can reference existing subjects if they already exist in dbGaP. This happens a lot with, say, like Coriell cell lines or DNA repository samples. The next tabular data that we want is this file of subject to sample mapping. And this is simple. It matches a study participant to any number of samples, because often with the GWAS, well, and as other studies, we have multiple samples per subject, and we want to keep those two concepts distinct because samples are processed in different ways, run on different molecular data types, different assays. So we need this table, and this can be updated separate from the subject listing. We also have a sample attributes file, which is essentially phenotypes for the sample. This could be things like what plate the sample was on, what method was used to extract the DNA for the sample, whether or not the sample is RNA or DNA, those kind of things. And then we have a core table, which is the pedigree information, which is the family relationships between the patients or the study participants. And this file is a core file, but it's not always required. There's a lot of studies, especially some of the early GWASs, where none of the subjects in the study are related. So this is only required when there are known relationships collected by the study. Now, I want to talk about the submission of the phenotype data files first. One thing, kind of in the broad sense, you want to compile the files. There's a study configuration, which acts as a study metadata file. There's study documents. These are the protocols or data collection instruments that are used. There's the core data files, which I just described. And then these files are transferred up to the submission portal. It's an online system that I'll show later, and that's done by the submitter. The data lands on protected servers at NCBI with very limited access. People in my group and others, like SRA, that can actually see these data once they come in, until they're processed and released through the controlled access mechanism. When the files come into dbGaP, dbGaP assigns curatorial staff to process, check the data, and bundle it for release. The dbGaP staff also does some basic sanity checking on the data to make sure that it's consistent with itself. We don't know truth of what the data is, but we do know if it's consistent with itself. And so those are the kind of checks we do. And I'll talk more a little bit about that as we go through this. And then we can send variable summaries back to the PIs for verification. We do that through our preview site mostly. I already talked about the phenotype data files or rectangular files. They're accessioned with a PHT number, so they could be referenced later if you wanted to talk about a specific set of data in a paper that you publish. Then you can do that. These rectangular files normally are one row per subject, and then each column would be a variable. But they can be longitudinal, such that you have a subject time point for each row. And when we have more complex tables like this, then we can't really show summaries available on the public pages. But if they're not complex and it's just a single subject per row or a single sample per row, we can show variable summaries on the public web pages. And then the dbGaP staff matches the data that is submitted back to what we call a data dictionary, which describes each column in the data file and any coded values that they may have. And I'll show more of that in a minute. I do want to point you back, if I could just back up to this. You'll see here there's a link to the submission guide. This actually goes to a zip package. And when you open that zip package, you see 22 items. Now that looks pretty intimidating, I think, when you first see it, but it's actually a lot simpler than it looks at first blush. The way the the submission package is set up is there's some general instructions, and these are the Word documents shown here, submission guide instructions. And I advise anybody that is submitting, the actual data submitter, to read these. It's quite verbose, but we have to cover a lot of bases because we see a lot of different types of studies coming to dbGaP, and they're all a little bit different. But this guide more or less covers every case that we've seen so far, and then it would be updated if we see a new case. We will actually update the submission guide to kind of cover that. This document kind of talks about versioning instructions, and that'll happen when you go to update an existing study, either redact or update or append data to it. Just to go through this submission guide package, the next file you probably should look at is kind of the study, what we call the study config, which is study level metadata. And this kind of instantiates the study in our system. And that's a Word template, which I'll show in a later slide. There's subject phenotype data. Now, there can be any number of these, and you'll see that there's actually two files here. One is a data set, so this is the data table with the rows are people, the columns are variables, and then the data dictionary, subject phenotypes data dictionary. And the data dictionary, each row describes a column in the data set. So, and then the same for sample attributes, subjects, sample mapping, pedigree. Now, the molecular data, so these are the genotypes, expression, or other types of data that you may have, is covered in this Genotype Submission guide, and so read about that. The thing about the molecular data in dbGaP, which might surprise some people, is this is actually the simpler data because a lot of these type of data are in established formats that we can process, and we process a lot of different formats of genotype data. But if you have questions, you can always contact us on the data format that you might prefer. The other type of data that's covered here is the association results, the analysis data. And these guides kind of show, this is a guide of how to format that data, and then this is an actual template that you can use. And then the rest of these files are used to link controlled access data back to public NCBI resources like GenBank, Geo, Trace, Archive. And why do we need these? Well, it could be that you have controlled access data for your genotypes and phenotypes, but you also have array expression data in Geo fully public. This mapping would allow somebody who has controlled access to do the link, but it would not, the data in Geo itself would not be linkable back to the data in dbGaP unless you have controlled access. So that's what those are for. I kind of wanted to just show some of these files from the submission guide. Here's what the study configuration template looks like. This is study metadata. As you see, there's kind of these, I guess, pink colored boxes show kind of instructions and give you examples. But this is where you enter the study name. We have two study names. One's a short name that we can show on Entree. The other one is kind of a longer name that would be shown on the study page. And you can see examples of what a study page looks like by clicking on, say, like this link. The type of study, molecular data that's in the study, you can have a study logo. If it's a named study that has its own logo, we can point out to a URL where that is. A description of the study, inclusion, exclusion criteria, how the participants were selected, ascertained for the study. We want to be able to index the study by phenotypic terms. If there's any genes that are specifically looked at in the study, they can be listed here. Articles that you or maybe, may have published on the data that's being uploaded could be listed here. Study attribution, important part, is where you would list your names and your colleagues, and then the funding source of the data. And then kind of a broad grouping of phenotypic categories that would facilitate people to search for your study later on when they're going to dbGaP to look for data. Another template that I wanted to show you from the submission guide, and this is a subject consent file. This is an example subject consent file. And so here we have the subject IDs listed. We have their consent. There's two consents here. One is 1. 0 is actually reserved for people that weren't in the study but are necessary for linking via the pedigree structure. And so I just wanted to show this isn't always the case, but we do have some studies where the person is referenced here in the subject file, but there's no molecular data because they weren't part of the study. They're just an aunt or an uncle that links a pedigree structure. So there's no consent. They're not released, but they are used to kind of build the pedigree within the programs that can use that. Also, so these two columns are the only thing required for this file. But there is a way to actually refer to existing samples that might be in dbGaP or later might come to dbGaP by using what we call subject source and subject source ID. And then for those studies where it's applicable, you can enter, submit the affection status. That would be more for a case control study. I wanted to show you the data dictionary, but I think my slide got messed up. Oh, here it is. Okay, so here's an example of the data dictionary for this data file. So as you see, each row represents one of the columns. And so subject ID, this is the de-identified subject ID that's submitted to dbGaP. Consent group determined by the DAC is how they listed it here. The source subject ID, and so on. And then over here you see where the coded values can be coded, so that the affection status says 1, 2, 1, 2, 1, 2, but 1 is control and 2 is case and 3 is other. I'll show more examples. Here's kind of a more detailed example of a very simple phenotype file and a data dictionary that would come into dbGaP. So here they have the subject ID, age of onset, height, birthplace, sex, race, education, and so on. And then those are described here in the data dictionary, and it points out to documents that they've uploaded for that relate to this variable, and other things like the data type of the variable, units, min, max, and then any coded values would be in this values. So these are the files that we're expecting to come in for a dbGaP submission. So once the data comes into dbGaP, the curators in my group, we check that the data submitted is consistent with the data dictionary submitted. And you'd be surprised how often there'll be a data value that comes in on a data set, and it's not described anywhere in the data dictionary what that value means. It's important for end users to have that, or they're going to be asking the question to us, and then we're going to be asking you again what it means. To avoid that, we try to do this ahead of time on our data checking. As I said, we look at the description and the units and make sure they make sense. You know, you wouldn't want a blood pressure of 2,000 or something like that. And so our curators actually have to do a lot of this by hand, but we have scripts to do other things like check for HIPAA violations and make sure the core tables match the phenotype tables. Like we might have in the pedigree file, this person is a mother according to the pedigree file, but when we look at the phenotype file, their sex is male. So that would be a check that we would catch, and we'd say, hey, wait, something's not adding up. Either the coded value is wrong or something else is wrong. We need to fix this. So that's kind of the iterations that we see when we're working with our submitters. And then we, any issues we see, we report back to the submitter, and we try to work those out and resolve them before we release to third parties. I just wanted to mention the document processing because I think it's kind of unique to dbGaP. I don't know of any other archives like dbGaP that do as much as we do on document. And the reason why is that NCBI has the infrastructure to do document processing through PubMed, Central, and so on. And so we actually like to have the original protocols and data collection instruments that were used in the study. And what we do, and you'll see it on our public website if you poke around, we link variables back to specific questions in a questionnaire, or variables back to protocols on how they were produced. And we do this both using vendors that we contract out, and then we do in-house markup to XML. And so this could be a benefit for you. We would actually mark up your protocols and data collection instruments in XML. And like I said, it allows the end users the ability to kind of figure out how the data was collected and what it means before they try to download it. I'm not going to go into a lot of the details on Genotype Submission, but I'll try to answer any questions at the end about this. But we have this Genotype Submission guide, which I showed you in our submission packet, and you can read about it. I guess the bottom line is what we tend to see mostly for the genotype submissions are formats like, we see a lot of Plink files coming in, which we can process no problem. We see, originally we saw mostly the vendor software information coming off of the Illumina and the Affymetrix chips. More increasingly, we see Plink nowadays. We also see formats like VCF and MAF, which is, what is it, mutational something format, that's used for tumor normal SNV calls. So once you've formatted your data and your study is completely registered, you'll want to upload your data, and the way to do that is through our submission portal. And as I said earlier, the data submitter or PI will receive an email invite for upload. Now, this invite happens kind of automatically once the study is completely registered. But I want to point out that this invite should be accepted within 1 week. Now, you don't have to submit within 1 week. You just have to accept it within 1 week. Otherwise, we'll have to reissue it. Once accepted, the access doesn't expire unless you tell us to. Just to remind you, that's kind of this process here would be through the submission portal. And then I wanted to just show you an example of the email that's sent out for the invite. It's kind of lengthy, but it has links to go on, instructions on how to log on, where to go, links to save, for future, and then down here you'll see that there's a help desk for any questions that you might have on the file submissions, and then any questions that you might have regarding the actual study registration. You would have your GPA listed here, and you could email them. Just to show you what the submission portal looks like, I've kind of blanked out. This is a real example. I had to blank out the names here. When you log on, you'll see that there's places to upload those files that I had described earlier, and then you just choose the file, browse for it, upload it. These are kind of the core files are shown up here. On this, I kind of scrolled down on this page, and now you see the pedigree file. You can be uploaded. Any number of phenotype files can be uploaded. We have some studies that are longitudinal cohorts, such as Framingham, that have many years of exams, and so each exam cycle in each cohort has its own phenotype file that's uploaded. So you can do any number of those. And then same with the molecular data. You can choose any number of those. Now, some studies are quite large, and so we have created a way, and this is only really for advanced users, to do a spare command line file upload, and then you'd use this link to do that. Once you've uploaded all the files, you don't want to forget to click submit, because it really doesn't happen until you do that submit. So you don't have to do this all at once. You can get the pedigree data together, add it in here, get the phenotype files, add them in. Then once you're all done and ready to submit, then you press submit. I'll also point out that there's kind of a history tab here that kind of tells us and you what's happened and when and by who. So once the data comes into dbGaP, there's an internal flow, and I won't talk too much about this, but just so that you know, there's kind of two pipelines in dbGaP under the hood. One is taking the phenotype and study metadata, processing that, doing QC checks, making sure they pass. If not, we have to iterate back. We want to make sure that the consent groups are matching what's registered and things like that. do the sample loading on the phenotype side and study metadata side. There's also molecular data is handled by another set of curators that are more kind of focused on that type of data, and they process it, do some basic QC checks, and then we do some sample matching and other types of checks to make sure that the phenotypes and genotypes are matching, say, for gender checks or relationships within pedigrees, first and second degree relationships are at least matching. Once everything's consistent within the submission, we start our process to kind of get things all ready and packed and loaded, and then we release out to controlled access for the individual level data, and then the public summaries go out on the web. And this is an old slide from our Nature Genetics paper back in 2007. It describes what we wanted to start doing. This was right before we released our first study, ARIDS, and we had already started a lot of processing on the Framingham cohort at this point. And so we wrote what we thought the process would be. And largely, this is still how we're doing the accessioning. And if you get a chance, I suggest you maybe read this paper. You can see what we were thinking 7 or so years ago, and that's still pretty much how we do things. Now, the policy originally was the GWAS policy. The new policy is the GDS, Genomic Data Sharing Policy. For the most part, that didn't really affect dbGaP that much, although there are a lot of things around the edges that were affected regarding things like submission certification, stuff like that, that we're working on. And we take all our guidance from the OSP and the data committee structures that they manage. But just this slide really is just to show kind of what we accession. We accession with a PHT, well, the whole study is accessioned with what we call a PHS number. And that's versioned, and we actually version the participant set if there's changes in consent and things like that too. We do the same for tables of data, so they could be referenced specifically within a manuscript. We also actually, each variable has its own accession also, and that's version too. Documents are versioned with a PhD number. Genetic data is bundled usually as large arrays of information that are accessioned with a PhG number. Analysis results, PHA number. And then there's other data that are linked through the controlled access SRA that have different accessioning space. That'll be covered more, I think, in a later webinar. At this point, I'd just like to kind of point out the people in my group that actually make dbGaP happen. There's a lot of other NCBIers, Steve Sherry, and many others, actually, to name, that are involved in dbGaP at a number of levels. Also, I'd like to say that it might surprise people, but there's actually more people working on dbGaP releases than outside of NCBI than inside of NCBI. That's because you saw the list of GPAs. That's bigger than this list of people processing the data. And the DACs are even more numerous because there's multiple people on each DAC. So I have to put thanks to them. And finally, I'll just say that, you know, none of the resource wouldn't be what it is without the cooperation of the submitting PIs and staff, and we thank them. And it's important, I think, for scientific data sharing. I'll leave it at that, and then I'll let Ben maybe manage the next part.
All right, great. Thanks, Mike. So we actually already have a ton of questions at this point. I have about 8 questions so far, and if you have other questions, please feel free to throw them into the question pod. So we'll get started, and we'll try to get through as many of these as we can in the next 10 or 15 minutes or so. The first question, this one was asked twice by 2 different people, and that is, can a PI delegate data loading to another individual on his or her research team or some sort of proxy?
Absolutely. That's built into our submission portal. I didn't show that part. Maybe I can get back to that part in the slides. Bottom line is in the submission portal, you have a way to invite people to, you can add submitters. So when somebody is originally invited, and that's either the PI or what we call the PI uploader assistant in the registration system, but you could add other people, and that's just done here once you've logged in. So the answer is yes.
All right, great. So another question we had, are there links between dbGaP and clinicaltrials.gov?
We have just started to establish those, so I don't know if I can bring one up on the fly, but yes, we have not a lot, but we do have a number of them, and now it's actually something new to our submission guide recently that we added a way for on the submission of the data you can reference a clinicaltrials.gov number. Usually that part happens before dbGaP. So we think we're probably in the better position to get the link from the submitters. But I think that answers that question.
Great. Okay, so there's two related questions here. Are there any required variables or phenotypes that need to be included, aside from subject ID, of course? And also, are there any standards implemented for data submission, like MinSeq or Miami, other things like that, or MIGS, for submission, in terms of common data elements?
Right, yeah. So our submission guide, like I said, it's very verbose. Each of those core tables that I mentioned does have a required element. So for example, in the subject consent table, the subject ID and the consent is required. That's absolutely required. Now there's the optional fields where you can reference to the sample name, aliases of the sample name and stuff. There are things like if you do submit a pedigree file, you have to provide all 5 columns, which would be family, person, ma, pa, sex. So read the submission guide for each file that you're creating. It describes which are optional and which aren't. For the most part, I think, at least for extramural studies, there's an expectation from the funding institute usually on a minimal set of phenotypes, and those would be normally, I hear other GP, I'm not involved in this process too much, but I think what I hear is that project officers expect the data that's being published, the main variables and covariates that are used in the analysis are submitted to dbGaP so other people can reproduce the information in your publication.
So speaking of publications, someone was asking, is there a way to update publication lists? A year, 2 years after submission, something like that.
Absolutely. We can update those kind of in-between versions. So you wouldn't have to update the data. So we wouldn't version the study. But you can just notify us that I want to add some more publications to that list. Those would be added.
Okay, great. And how would somebody go about doing that notification?
Well, so like I said, when you upload data initially, you're assigned a set of curators, one from the phenotype pipeline and one from the genotype pipeline. Phenotype curator would normally update the publication list. You just contact them.
Great. Just a clarification question. Somebody asked, do we have to submit the raw data to SRA and VCF files to dbGaP on the same NGS dataset?
I would say you don't have to do both, but I think it's highly desirable to do both. The SRA data is not as useful to as many people as the VCF file, just because of the sheer size and computational effort that's involved in calling variants. But I think we at dbGaP have to defer to the program officers and GPAs on what their expectation is. And with intramural scientists, I think that's something you should discuss with your GPA on what you think is best. But I would say from a data sharing point of view, I think both would be highly desirable.
Great. One more clarification question, and I think you addressed this, but somebody said that the invitations to submit expire quickly. Is that true, or is that maybe a misconception?
Yeah, it is. The invitation and I think I put it on this slide, to submit, the invitation itself should be accepted within 1 week. If not, we have to reissue it. And the reason why we do that is we don't want these invites to be out in the wild for too long. I mean, it's really following the model that was set up by the manuscript submission process with PubMed Central. So there's a week to accept it, but it's not difficult to accept it. And then once you've accepted, you can log on anytime after that to do the upload.
Great. Thank you for clarifying that. So also, could you describe the release schedule for uploaded data?
Okay, so I think there's an expectation within the GDS policy that says 6 months after the data is produced. That's been somewhat interpreted on when the data is actually produced. Has been interpreted. It's like, it's a little gray on when exactly that is. Is it when it comes off the machine? But my understanding is most are interpreting that as when the data is QC'd by the group producing the data and ultimately submitting it. And so the QC process does take some time, and that's not really included in that 6 months. The 6 months is because there's no embargo in the GDS policy. So previously in the GWAS policy, we would release the data, but it would be embargoed for some time before anybody could publish on it. With the GDS policy, there is no embargo, but there is a protected set of time for the data producing researcher to process, and publish on his own data.
Great. So cell line data, should it go to dbGaP or SRA? I know that's not an easy question.
Well, I think you'll have to look at the GDS policy site. It actually talks about that. And it depends on when the cell lines were collected. So you have to pay attention to that. And I think if you work with your GPA, you'll be able to figure out what's appropriate. Oh, I just wanted to show you that if you're a researcher, you'll see in the data, institutional certifications are actually set up so that for studies, so that extramural investigators have this section, intramural investigators have this section. But you'll see that there's a time frame that kind of changes what submission certification you use. If the cell lines were clinical specimens collected after August 31st, 2015, you'd use this one. Before then, you would use these two. So take a look at this. This might help. I can't really read the questions right now.
Right, no worries. And just to remind people that the SRA database is the unrestricted side of dbGaP. So you do not need authorized access if assuming that you have a sample that does not need to be protected under U.S. law. And I think with that, we're going to go ahead and wrap up the webinar. These questions and their answers should be echoed on the FTP site in the matter of a couple of days, whenever Peter Cooper gets to putting them up. And I'd like to thank Mike for giving this presentation. Thank you very much for coming, your interest in dbGaP, and for sharing data with the community.